Hello students, I am Masha Rodriguez, Assistant Professor in Botany from Carmel College of Arts, Science and Commerce for Women. Unit number 2, Plant Tissue Culture Technique, and the name of the module is Establishment and Incubation of Cultures, and this is module number 10 of Unit 2. The module outline: ideal conditions for incubation of cultures, setup of culture room, establishment of in vitro cultures. These are the learning outcomes of this module. Lists the ideal conditions required for in vitro cultures, describes the setup of the culture room, explains the establishment of in vitro cultures. The steps in plant tissue culture technique are as follows: preparation of culture media, preparation of explants for in vitro culture, establishment and incubation of cultures, and maintenance of cultures and subculture. Students, in the previous module, I explained to you about preparation of explants for in vitro culture, that is the selection of explant, isolation of explant, surface sterilization of the explant, and finally the inoculation of this explant on the nutrient medium. In this module, we will look at the third step in plant tissue culture technique, that is establishment and incubation of cultures. The last step, that is maintenance of cultures and subculture, I will teach you in my next module. Students, in this slide you can see an image, and in that image there is a culture vessel which is inoculated with seeds. So in the previous module, we had the last part, we had learned about inoculation of explant. So here we have a culture vessel which is inoculated with seeds. Now culture vessels, as I told you in the previous module, can be a bottle, or it can be a petri plate, or a test tube containing nutrient media. And here we have a culture vessel, that is a bottle, and it is inoculated with seeds. Now this culture vessel, or which is inoculated with seeds, this explant that is inoculated has to be given the ideal conditions to grow. So this bottle, that is the culture vessel along with the inoculated explant, has to be transferred to another room of the tissue culture laboratory, that is the culture room, where all the growing conditions or the ideal conditions that are required for this explant to grow are provided. Before we move on to the culture room, we will first learn about the ideal conditions for incubation of cultures. So the three ideal conditions that are required, or the physical environment that is required to support or to facilitate incubation of cultures are: the first one is temperature. The temperature has to be 25 degrees plus or minus 2 degrees. Second is the photo period, that is the amount of light that is required for the explant to grow. Normally, 16 hours of photo period is given to the explant. And the last is the relative humidity, that is around 60 to 80%. Now this relative humidity is maintained within the culture vessel because in the nutrient medium there is water that is present. In this slide, you can see an image that is the culture room, and you can see on all the three sides there are racks that are kept, and they are painted white in color, and each rack has got a fluorescent tube, and the cultures are kept on these shelves. As I said in the previous slide, three ideal conditions have to be provided, that is temperature, photo period, and relative humidity. The temperature is regulated by using an air conditioner, and that's how you provide 25 degrees plus or minus 2 degrees. Next is the photo period. That is how. Next is the photo period, wherein artificial illumination is provided by fluorescent tubes, and these fluorescent tubes are placed for each shelf. The third one is relative humidity. As I said in the previous slide, relative humidity has to be around 60 to 80%. In case if there is any excess amount of humidity that is present in the culture room, then this excess humidity is taken out by the air scrubbers. And yes, the culture room has racks or shelves on which the culture vessels are placed. In case there is liquid culture, then these liquid culture, which is in the flask, culture flask, has to be placed on the rotary shaker because liquid cultures require aeration. Now we will learn about the establishment of in vitro cultures. So once the explant is inoculated on the nutrient medium, then that culture vessel is then brought into the culture room wherein all the ideal conditions are provided, such as temperature, the photo period, and the relative humidity. Photoperiod depends upon the type of the explant that is used. Some explants require less amount of light, some require darkness for growth. Uh, in case of callus, it requires a dark period followed by a photo period. During incubation of cultures, there are certain events that take place. The first is absorption of the nutrients. As the explant grows and as you provide the ideal condition, the explant starts to absorb the nutrients that are there in the nutrient medium. And as nutrient absorption takes place, the cells begin to enlarge, they increase in size. Cell enlargement takes place and cell division takes place. The cells begin to divide, and finally, there is callus formation. Callus is a mass of undifferentiated parenchymatous cells. Students, in this slide, the first image is the inoculated explant. Now, as I told you, the culture vessel can be a bottle, it can be a petri plate, or it can be a test tube. So here we have the explant which is inoculated on a petri dish containing the nutrient medium. And after 3 to 4 weeks of incubation, we will see the growth of callus tissue. So the second image is a callus tissue, that is the undifferentiated mass of parenchymatous cells. So students, in this module, we have learned about the establishment and incubation of cultures, wherein we learned the ideal conditions that are required for incubation of cultures, that is the temperature, the photo period, and the relative humidity. We also learned about the culture room, the setup of the culture room, and all this ideal conditions that are there are provided in this culture room. The culture room also contains racks or shelves and a rotary shaker. And finally, we learned about the establishment of in vitro cultures, wherein different events take place during incubation of the cultures. These are the references that I have used to prepare this module, that is plant tissue culture by Mishra and plant tissue culture by Singh and Srivastava. This is an additional reference, that is a web link. Thank you, students.